and how we investigated a membrane in cochlear aqueduct and used virus-mediated gene therapy to rescue hearing. And then at last, I would like to discuss the future directions or come up with some ideas on that. So to understand why the glymphatic system is important, so this is fluid flow in the brain, we have to discuss, or we have to understand the neurodegenerative diseases. And here is the example of Alzheimer's disease. So Alzheimer's disease is characterized by genetic and environmental factors that lead to amyloid plaques, which is clusters of amyloid beta protein in the brain, and also these tangles of tau protein, leads to neuronal disruption of physiological functions, resulting in the memory loss and cognitive decline that we see in Alzheimer's disease. So my supervisor, Maiken Nedergaard, and some others came up with the hypothesis of the glymphatic system. So the glymphatic system is a hypothesis that the cerebrospinal fluid goes down to the periarterial spaces, washes away waste products like the protein amyloid beta, and then washes it out to the perivenous spaces, to the periphery. So in the glymphatic system, the glia cells is very important, and it's like the lymphatic system in the rest of the body, so therefore the glymphatic system. So when the fluid is washed away, it goes out through the cribriform plate, the cranial nerves, the spinal nerves, and the meningeal lymphatics to the cervical lymphatics and to the rest of the body. So the glymphatic system is most active during sleep. So this is a study by C et al. Published in Science in 2013, where they inject tracers during awake and during sleep. And we see that during awake, which is the yellow here, we see that there is the tracer is not distributed as much as during sleep, which we see in green here in the brain tissue. And when we when they quantify it, we see that in sleep there is a much larger area covered by tracer compared to awake. So they also injected amyloid beta, which was the protein accumulated in Alzheimer's disease, and see that the rate constant, so the speed of which the tracer is removed from the brain, is higher during sleep and during KX, ketamine xylazine anesthesia, compared to during awake. So in the glymphatic system, it's very dependent on the aquaporin 4 channel, which is situated in the astrocytes. So here is an experiment where they inject a MRI tracer into cisterna magna. And in wild-type mice, we see a nice spread of tracer. But when we have the aquaporin 4 knockout mice, we don't see the tracer spreading as nicely. So we have now learned that the fluid flow in the brain washes away waste products. The glymphatic system is most active during sleep, and it's dependent on the aquaporin 4 channels in astrocytes. But today we're here to talk about the ear. So I know most of you is probably much better at both anatomy and physiology of the ear than me, but I'll just briefly go through it. So when we hear, sound waves comes in to and it's transduced by the ossicles in the middle ear and to the inner ear. In the inner ear, we have the cochlea, which is perceiving the sound. And if we make a cross sections, we see the 3 compartments. So we have the scala tympani and scala vestibuli with perilimbs that are similar in composition to the cerebrospinal fluid. And in scala media, we have the endolymph, which is completely different. And when the hair cells bend, the signals is transduced via the spiral ganglions to the brain. So when I started in the lab, Maiken and the people working in the lab has just found out that when we injected tracer into cisterna magna, we saw it entering the ear. So this is a brain seen from below, where we have the two ears. You don't see my cursor. So the two ears up there, with the covered in the white circle, and when we put in tracer, so the tracer come in here in the subarachnoid space and into the inner ear. And we see that within, so this is 1.5 hours of scanning, the tracer first comes into the base of the cochlea, and then as time goes, we see it coming in towards the apex. We also see that it only comes in one of the compartments, the scala tympani, and not scala vestibuli here. And we see that there is this channel-like structure where it comes in through. So we looked in the literature and asked the old professors at Copenhagen University, and they told us about the cochlear aqueduct. And the cochlear aqueduct is a channel from the subarachnoid space to the inner ear. And we found some old papers showing that if it's closed, we see protein accumulation in the inner ear, which led us to think maybe it's similar to the glymphatic system, that it's important for washing away waste products in the inner ear. Then we also know that infections can spread through the aqueduct, and it gets inflamed at meningitis. I'll come back to that. So then we looked further into the images in the MRI that I just showed before. So here we quantify the tracer influx. So first we see the tracer in cisterna magna, the green, and then we see the tracer in the subarachnoid space just outside of the inner ear, then in the cochlear aqueduct, and at last in the inner ear. So the MRI has the advantage that it's very high temporal resolution, but not so high spatial resolution. So we also did the same in the CT. So here we have the possibility of making 5 RIs within the cochlear aqueduct. And we see, so here the darker color represents closer towards the subarachnoid space, where the lighter colors represent longer away from the subarachnoid space. And similar in scala tympani, the closer we are to the cochlear aqueduct, the darker the color. So we see that the tracer first rises in the beginning of the aqueduct, and then later, and to a lesser degree, in the end of the aqueduct towards the inner ear. And we see the similar pattern in scala tympani, but it doesn't reach the apex within the 30 minutes of scanning we did in the CT. So, because we were a glymphatic, or we are a glymphatic lab, we wondered, is it aquaporin 4 dependent? And we saw that, or we injected in cisterna magna tracer in aquaporin 4 knockout mice and wild type mice, and did the MRI scanning and looked at the tracer distribution. And we didn't find any significant difference in indicating that the flow into the inner ear is not dependent on aquaporin 4. Then we did some modeling, and we used the diffusion equation. And here we have the location. So 0 is close to the subarachnoid space, whereas 300 micrometers is close to the inner ear. And we see that the model fit the data very nicely. And when we compare the diffusion equation, or the diffusion constant that we optimized for in the modeling, we see that it's slightly higher than the theoretic diffusion coefficient based on the Stokes-Einstein equation, indicating that there might be some kind of dispersive mechanism helping with the flow or the transport of tracers into the inner ear. So then we asked, is there free passage of molecules from the subarachnoid space to the inner ear? So while we did the project of the inner ear, some other people in the lab did a project on the subarachnoid lymphatic-like membrane. That is a new membrane, or recently discovered membrane that divides the subarachnoid space and is important for fluid transportation and lymphatic function in the brain. So then we looked for it in the cochlear aqueduct. So here we have the scalar tympani in the bottom of the images and the subarachnoid space in the top. And we see that this membrane actually goes down to the side of the aqueduct, forms a membrane, and goes back on the other side. And it expresses lymphatic-like markers and is continuous with the slim in the rest of the brain. So then we wondered, does it have some kind of function, this membrane? So we injected 0.2 big microspheres into the brain and saw that the membrane restricts the transport. It seems like the microspheres get trapped by this membrane, but within 24 hours, there is some microspheres that reach the inner ear when injected into cisterna magna. So it constricts or restricts the flow, but it doesn't block the flow. Then, as the slim in the brain is active, has lymphatic activity, we also looked for lymphatic cells. So here we see this is a zoom in where we have the end of the membrane here. And we see that there is macrophages that are LIVE1 and IBA1 positive attached to the membrane inside the cochlear aqueduct. And when we looked in the animals injected with microspheres, we saw that the macrophages actually engulf the microspheres, indicating that they have some kind of lymphatic function blocking the transport into the inner ear. So then we asked, if microspheres can come in, what about virus? So we started injected green virus in cisterna magna and hypothesized that it came in to the same direction via the subarachnoid space, the cochlear aqueduct, and into the inner ear. And we saw that the expression pattern was consistent with this hypothesis that the virus comes in to the inner through the scalar tympani in the base first, and then in the middle, but not so much in the apex, similar to what we saw in the other data. And when we compare with IV data, so intravenously injected virus, we don't see this pattern, and we see the expression to a much lower degree than what we see when we inject in cisterna magna. So now the big question arises, can cisterna magna injection rescue hearing? So I know most of you know, but to describe it, we used Wiglet-3 deficient mice that are deaf, and it's a mouse model of the DFNA 25 and autosomal dominant and progressive sensorineural hearing loss. And what happens in these mice is that the vesicles, the glutamate vesicles inside the hair cells, they get decoupled from the receptors, and then the signal cannot be transduced into the brain. So if the mice is hearing, we see this wavy pattern of the auditory brainstem response. Each wave represents an anatomical location in the inner ear. But if it's not, we have this flat line. So when we have Wiglet-3 transductions, we see that the vesicles in the hair cells and the receptors on the neurons, they get coupled, and thereby they can talk to each other. So our experimental setup was so that we did a hearing test 1 to 3 days pre-injection. So this was done in adult mice. They were about 2 months old. And then we did a cisterna magna injection of either virus or ACSF, which is a sham injection, so artificial cerebrospinal fluid. And then 2 weeks later, we did a second hearing test and euthanized the animals for histology. And luckily, the mice was hearing. So here we have a wild type mouse. We see the nice wavy pattern. We have a knockout mice injected with ACSF, and we see that there is no response when a sound is played for the mouse. And Then we have the mice that were injected with virus, the knockout mice that were injected with virus, and we see that this wavy pattern is restored. And when we look at different frequencies over here, so here is the animals that were injected with ACSF, and we see that both before, which is the open circle, and after the full circle, so before and after injection, they are deaf, and the wild type, they are hearing. But when we give them the virus before injection, the animals are deaf, but after the injection, we see that the mice actually get hearing with similar threshold to wild type animals. Then we also looked at the histology. So in the wild type, we see expression of VGLUT3, which is in white here in the images. In the knockout, we see no expression of VGLUT3. And when we inject the virus, we restore the expression of VGLUT3 in the inner hair cells. And what I find very, very fascinating is that we can also see the restoration of the architecture of the synapses. So here we have a wild type mice. The vesicles and the receptors are coupled. They can talk to each other. And knockout, where the yellow and the magenta are not coupled, they are decoupled, cannot talk to each other. And then we have an animal that were injected with virus, and we see that this architecture is restored so that the receptors and the vesicles can talk to each other and thereby transduce the signal. So, of course, we have some limitations. So we have some ectopic expression of virus. We also have the problem of the cisterna magna injections. We don't see full recovery of the amplitude of the auditory brainstem response. So in order to address some of the limitations, we looked at the inflammation upon injection, both in the brain. So we looked at the perimeter of the IBA1 expressing cells, so that's the microglia. And we saw that it was not different between this is wild type animals injected with the virus or with ACSF. We also saw that the inflammatory markers in the inner ear was not upregulated in the animals receiving ACSF, receiving virus. And then we also looked at the behavioral response. So there was no difference in weight. And we did an open field test where we put the mice in a white box and looked how much did they move and how quickly did they move, and saw that there was no motor function impairment. And also this center in time versus periphery, which is a measure of anxiety, was not changed within the two groups. So as future directions, so as I said in the introduction, or as was said in the introduction, I'm working on modeling flow through the aqueduct and thereby maybe optimizing transportation. We could also work on other therapeutic agents and minimizing side effects, maybe by working on specific viruses or injection in the lumbar region instead of the cisterna magna, because that's much more widely used in the clinic. And then, so as I said, or as some of you might know, I work in a general practitioner now. And in the clinic, there is this small little girl. She's 4 years old. And 4 months ago, she had meningitis. And now she has a cochlear implant on one side. And given the project or the histological data on this membrane in the inner ear with the macrophages, I thought it would be very interesting to work on whether we can somehow know more about the spread of immune cells between the brain and the inner ear. So I'll just tell you a little bit about the modeling that we're working on now. So we're working on diffusion versus advection. And then we're also working on this spatially variant diffusion coefficient. So over here we have 0, it's close to the brain, and 300, so it's the distance. That's in scalar tympani. So we see that the diffusion coefficient seems to be lower in the cochlear aqueduct, maybe representing that this membrane blocks the diffusion inside the inner ear, whereas it rises in the scala tympani where there is no membrane. So, in conclusion, tracers injected into the cerebrospinal fluid reach the inner ear. A membrane in the cochlear aqueduct is continuous with the arachnoid and express lymphatic markers and restrict transport into the inner ear. So, and systemic magnet injection of gene therapy can restore hearing in adult mice that are VGLUT3 deficient. So, I have, of course, not done all of this myself. So, this project is a collaboration between my lab with Maiken Nedergaard leading, the Karolinska Institute with Barbara Cameron leading, and Harvard Medical School with Jeffrey Holt leading that group. And then I would like to give a special thanks to Leo, who is the first author on the paper and have done a lot of the experimental work while I was in medical school. And also Peter Borg, who is the one who brought me to the lab and has done a lot of the scientific discussion with me how to move forward on the project. So thank you for listening, and time for questions. Oh, just
So do we have any questions? Oh, we got a couple here.
I think I have a very loud voice. So does your imaging data suggest that there is no connection through the helical tremor for the parallel length?
So the problem with our imaging data, I'll just so the problem with our imaging data is that we only scan for maximum 1.5 hours. And here in the CT, where we have the very high resolution and the ability to make our eyes in the scala vestibuli, we only have a scanning of about half an hour, which is, I think, too little to be able to have the tracer all the way up to the helicotrema and then back. But when we look in the video, which is 1.5 hours of scanning, we see that the tracer reached towards the apex of the inner ear. So I think it's mostly a matter of the time that we look on, and not so much that it doesn't go up there.
Do we have any questions? We got time for.
Oh, hi, great talk. Do you have any idea of the size of the molecules that can go through that membrane through this?
Yeah, so we actually didn't test that bigger molecules that much bigger. So we have, we did test 1 micrometer big, and they also are able to get through. So, but not bigger than that.
Thanks.
Yeah.
It's spectacular. So I learned in training that the cochlear duct closes as the patients get old in people.
Yeah.
So I don't know if that's true. I mean,
yeah. So there is
so
discussion.
is that true in mice? And how does that affect this?
Yeah. So there is discussion about that also in the papers describing the humans. So it's not completely sure that it does close. There is data on old adults where it's still open and also where it's not. So it's not completely well established whether it's closing or not in humans. So in mice, I don't know, unfortunately, whether it closes or not. But yeah.
And how would this compare, this mode of transport for therapy, compared to, say, going through the round window or into the lateral canal?
Yeah, so when we inject into systemic marker, we have the benefit of getting both ears at one injection. So that's also actually one of the points discussing with some of you yesterday, that it's a quite easy injection in mice. So it's something that's accessible to do, so it can bring the field forward because you can reach both ears in a mouse pretty easily. So to answer your question on to compare, so we get both ears, which is nice, but we also get infection, or we get the virus into the brain as well. So we also get that when we inject into the inner ear because of the aqueduct. So the virus also gets into the brain in that injection, but we have a higher concentration in the ear when we inject directly into the ear compared to the cisterna magna injection. But what I would think, moving towards translational aspect, I think it would be nice to see if we could make it in the lumbar region, because that's much less, it has much less dangerous structures next to it. Yeah.
So with that, we've had an absolutely fantastic presentation and some fantastic discussion. I would like to invite everyone to give another round of applause to Dr. Maiken. So let's get ready for the camera. This is absolutely a brilliant presentation. Thank you so much. It's wonderful data.
Thanks a lot.
Yeah. Great.